好，谢谢 Frank，还有感谢 Tyra 给我这个机会来跟大家分享我们实验室过去3年在做的一个很特别的胶水凝胶。然后因为嗯语言的关系，我可能还是会用英文来报告为主，然后之后问问题我们可以用中文。So yes, hi everyone. I would like to share about this special jelly that I have been working on throughout my PhD and master. The title of my talk would be Dynamic Matrix with DNA-encoded viscoelasticity for cell and organoid culture. To set the stage, I want to first highlight that we know that human body contains 70% of water, but if you pinch yourself, your skin or your muscle does not flow like water. This is because there's a extracellular matrix, a jelly-like material that hold our cell and tissue together in the 3D structures. This extracellular cellular matrix contains collagens, laminin that you might heard of in cosmetic products. But essentially, those are protein polymers that provide structural support for cell to grow in 3D manners. Well, it's kind of like jelly, but in our body, it's more than just one component. There are several components playing the role. And actually, extracellular matrix takes up a big part of our organs and tissues. For instance, on the right side, you see a pig kidney. If we remove all the cells from this kidney, then we left on a kidney extracellular matrix. And you can see that it's a kind of translucent jelly-like material that actually plays a major structure of the organ. And this concept has set the direction of the research, transforming from 2D cell culture to 3D cell culture, since our body are in 3D structure. So there are several materials that want to mimic this extracellular matrix and provide an artificial platform for tissue modeling. For example, you can culture cells in the matrices, study the cell's natural behavior in the lab, or you can even further develop organoid, which is a miniaturized organ model that simulate partially the function of a real organ in your matrices. 3D cell culture is a powerful tool for many different applications, ranging from basic research on understanding how cell and tissue develop in your body, or you can further develop a disease model that can be used later for drug screening, which could further develop into a personalized medicine that worked for individuals, patients. However, most of the 3D cell culture studies still rely on matrigel, which is extracted matrices from mouse tumor cells. They have multiple problems, but I think the major problem is that this material lacked ability to reproduce a realistic mechanical environment. What do I mean, realistic mechanical environment? If we look at biological tissues, it is actually not a simple elastic or viscous material systems. On this graph, you can see on the x-axis, we have the stiffness or the elasticity called G prime. On the y-axis, it's called viscosity, labeled as G double primed. You can think of honey like very viscous fluid solutions that has the G double prime dominate over your G prime. And on the other extreme, you can think of like gummy bear as like a very purely elastic solid. And actually, our biological tissue lies between these two extremes. It shares the characteristic from both type of material. This viscoelastic material has a typical behavior called stress relaxation behavior. If you think of like pressing this gummy bear, and this gummy bear, of course, will have a resistant force that push back your forces. And however, this force could decay over time. So essentially, stress relaxation behavior on this graph is a stress decaying over time between different tissues. Gummy bear probably have a very, very slow decaying rate, very, very slow stress relaxation rate. And honey will be the fast example of it. If you look on this graph, you will see that different tissues shows different stress relaxation time. It decays at a different rate. And this is quite important because this plasticity allows the cells to push the matrix when they're growing in 3D. And at the same time, the matrix also push back. You can imagine cell will have trouble to expand or divide it if the matrix it doesn't allow any relaxation, any space for the cells to push against it. So you will want some viscoelasticity to some degree, not too much, just the right amount, that allow the cell to change their shape, expand it, but still retain in the 3D structure. This tuning the stress relaxation property have a large effect on cell development. You can see from this result from previous literature, they found that fibroblasts in the alginate hydrogels grow different morphology when the relaxation time is become faster and faster, more fluid-like. I want to highlight here that the problem of these previous systems is that it's difficult to change stress relaxation without touching other composition like the polymer length or the charge of the polymer. Essentially, that change the chemical composition that might have other effect on the cell together with your stress relaxation effect. Ideally, a cell culture material should have a programmable and dynamically adjusted property to fulfill the need from diverse cell types. I want to take a little bit of time talking about what exactly are programmable material. In your classical polymer materials, usually we describe it as that it responds to a stimuli and give you just only one response. For example, like a rubber band. No matter how many times you stretch it, it will just elongate it, become longer, and nothing will change it. However, a truly programmable material have three key characteristics: modularity, predictability, and abstractions. The molecular program in this material must be able to add it and replace in a modular manner. And this molecular program will produce a predictable response as dictated by the abstract reasoning. Now, there is a miraculous molecule that actually fulfills all three of these conditions, and that is, of course, DNA. We all know DNA as a universal genetic material, and it has also some quite remarkable property. In human cells, each cell has 3 billion base pairs. If you stretch all of this DNA, you will result a 2 meters long string, and also carry a large informations that will determine the cell fate infinitely. However, in DNA nanotechnology, we took it out of the biological context and use it as construction material. So whatever I just say, just forget about it. It doesn't matter for DNA nanotechnology. We care about how we can use it to construct self-assembled nanomaterial. This idea was first brought up by Ned Seaman, which he think of DNA strand self-assembly as building a Lego blocks. And this is like a picture of his office. He has been known for a very messy office. If you want to ask for some paperwork from him, you'll probably not get it. Anyway, he pointed out two important features of DNA. The first feature is the DNA binding is highly sequence specific. I trust everyone here pay attention to your biology course in high school, that you know that DNA consists of 4 different nucleotide bases, A, T, C, G. and A always binds to T, C always binds to G. So by looking at this sequence on the slide, you can predict that which strand will perfectly complement to this sequence. It would be G, T, G, A, C, C, and so on. Because of this selective binding, this makes DNA uniquely suited for programmable cell assemble material. This is a big difference from other type of material which do not follow this type of predictable behavior. And DNA nanotechnology leverage this property to build custom, for example, custom nanostructure by combining multiple different DNA strands, and they can bind in a predictable manner. And you can build this like a DNA origami, like this smiley face. You can also design a DNA machine that essentially use a lot of strand that undergo strand displacement and can design it to do certain jobs. But our group actually more interesting in building macroscope in DNA material. By combining classical polymer with a DNA, we usually like to use this analogy, like polymer is a screwdriver and DNA is like this tip. When you change the tip on your screwdriver, you also change the function of how you can use this screwdriver. So the central question around my project is, can we harness the power of DNA nanotechnology to precisely control the hydrogel property? And of course, the answer is yes, we can do that. When classical polymers combine with DNA as crosslinker, something unique happens because DNA sequence carry information on how this crosslinker will behave. By changing the sequence, you can change, for instance, the melting temperature of this crosslinker, the binding kinetic, the mechanical stability of this DNA duplex. For instance, you can use force to rupture this DNA crosslinker and it will melt. But it can also self-heal, or you can increase the temperature to melt this double-strand DNA, but reform after you cool down the temperature. This process is reversible. An even cooler thing is that nature actually offers a massive toolbox for manipulating DNA. For example, DNA enzymes. You can cut DNA by using restriction enzymes. You can ligate them by using ligase, or even create nick by using nickase. There are other biomolecules that also and change the DNA property. For instance, protein, metals, or ions. Another big part in DNA nanotechnology is actually called dynamic DNA nanotechnology. We utilize strand displacement. What it means is that basically you add another strand that invade original DNA strand, replace it, basically kick out the original strand that bind with it. And this replacing strand is more stable, so they are in favor of binding. But you can also do this multiple times by replacing with a more stable strand. This is a powerful technique to induce transformation on DNA complex. I would not talk much about this part as my DNA hydrogels didn't use much of this technique. Of course, we're not the only one who come up with the idea of using DNA in hydrogel systems. Some study use DNA as a whole material component, that's all DNA hydrogels, basically connect double-strand DNA with some sticky end, and they will form a very stiff extending gel structure over here. Another study, similar to what our idea, using DNA as crosslinker in the polymer material, and they induce this crosslinker conformational change to change the shape of the hydrogels. But surprisingly, there are very few studies that have used DNA hydrogels as a 3D cell culture matrices. Even if they do the cell culture, they only show very shitty data, basically. And the main reason is that most of the previous report systems require a large quantity of DNA. this synthetic DNA you bought could be expensive to use at scale if you use a lot. And imagine in cell culture, you want to multiplex this, that would be prohibitively expensive to actually use in translational science. And if you think about it, at one point, if you bombard the cells with lots of foreign DNA, it could interfere with the cell and trigger some undesired reactions, like inflammations. So to address this, we set the goal to have, can we somehow use as little DNA as possible, but still have full control on the material property? Our strategy has a trick at using an ultra-long polymer chain as the material backbone. This synthetic material basically built from polyacrylamide called acrylic acid. And this synthetic polymer is tunable. You can change the charge or the molecular weight of the polymer. And the unique part also is that we polymerize with single-strand DNA. There's this anchor single-strand DNA on the backbone, which allows other DNA crosslinker to sit on it and form crosslinks. The molecular weight of this polymer is in the megadalton range, which is quite heavy, because we hypothesized that if the polymer is such heavy, then we can use less DNA crosslinker to form a firm gel, because there will be a lot of entanglement that support the whole structure. By adding DNA crosslinker, they undergo self-assembly, then we can form cross-linking matrices. This polymer has only been used in solution before, so our first goal is to establish a reliable cross-link to form a gel-like material. At the beginning, we'll say, okay, let's just throw some DNA inside and cross-link them. How hard could it be? We initially had a very naive design that just add a simple splint DNA strand that is complementary to the anchor strand. So it will bind to the two anchor strands on two polymer chains, and then ideally it should form a gel network. However, we never get a firm gel out from this strategy, and we realized that actually most of the crosslinker didn't play a role. It just hugged themselves, binding to the same anchor strand on the same polymer, and form this blob. This phenomenon is not only happening in DNA material. It's actually a universal phenomenon, typically happen in polymer material. For example, it's polyacrylamide-based acrylamide hydrogels. Most of the crosslink actually just form a loop. But since these materials are cheap, you can just dump a large of crosslinker to overcome it. Since our goal is to keep DNA crosslinker as low as possible, we need to come up with a more efficient way of crosslink our material. Then we later come up with solutions that use a splint pair. This splint pair has two domains. One domain that's complementary to the anchor strand. So when you anneal them on the thermocycler, they first will bind to this anchor strand, sit on it. The trick here is that this overhand domain, we purposefully make it very diverse. There are a lot of different sequences inside, so that when they start searching for the perfect match, they have less probability to find their match on the same polymer strand. So we can avoid this intramolecular loop bonding, which does not contribute to our stiffness at all. And therefore, we can enforce this intermolecular crosslink to happen, which is the effective crosslinks in this case. I want to highlight that how we design this diverse sequence, because in principle, you can just design 100 different sequences, do 100 different synthesis. But this is what makes your cost also quite high to not affordable in cell culture. So we use a combinatorial approach by introducing a random basis inside this overlap domain. So imagine there's an N base, which could be A, T, C, or G. If you have one N base, meaning that you will get 4 different sequences in this library, If you have 2 M-base, you get 4 times 4 will be 16 different sequences in this library, and so on. In this case, we only need to synthesize 1 tubes, but we can still enjoy a diverse pool of sequences. The nice thing about DNA is you can do simulation on it. In this plot, we want to know how this combinatorial affect cross-linking efficiency. This plot simulates what is the probability of one cross-linker will find the perfect match on the same polymer strain. On the x-axis is how many different sequences you have in your library, and the y-axis is how many binding sites per polymer chains. And ideally, you want your material to form in this blue region to have as low intramolecular crosslink as possible. There's a loop, less loop in here. And you want to avoid forming a gel at these red regions. So we pick a few conditions and do the experiment out of it. So it's shown in this white dot. We do annealing of our polymer with our DNA crosslinker, and we characterize its mechanical property by using oscillatory rheology. Some of you might have used this method before, but for the people who don't know, it is a method that characterizes the elasticity and the viscosity of your material. By putting your sample, sandwich it between the cone and plate, and this cone will rotate, shear your samples, and then it would detect the stress change on the different conditions. The most important part is if it's a gel-like material, the storage modulus representing the elastic portion of the material will dominate over your loss modulus G double prime. If it's a liquid material, then it's the opposite way. Your loss modulus G double prime will dominate over the G prime. And by this, we anneal our gel, we do our rheology, and this combinatorial approach worked out quite nicely. As you can see on the slide, when we don't have any cross-linkers, just the polymer solution, it looks like just a slimy solution on the glass slide. But if you have one spin library, then you somehow get a gel-like structure. With increasing number of spin strand in your library, and we get a stiffer and stiffer gel because we suppress this intramolecular loop formations. Again, I'm showing the simulation data, and the experiment result actually match nicely with our simulations when by increasing the splint diversity in the pool, we're increasing the stiffness over time. And I want to point out that we also discovered that if you have too many splint sequences in your pool, the kinetic of them to find their perfect match will become very, very slow. So at the end, you did not actually increase the stiffness because they just couldn't find a match in a certain amount of time. At the end, we found a sweet spot around 64 splint pair. But nevertheless, all of the gels use the same amount of DNA. And this combinatorial approach represents a unique way of controlling matrix elasticity without changing the cross-linker concentration. Simply by changing the cross-linker diversity, we can change the stiffness from 40 to 140 Pascal, and this actually is comparable to what matrigel can give us this range. And we're happy to reach this range. Furthermore, we're also happy that thanks to this complex library systems, we can achieve gelation at exceptionally low DNA content compared to other previous reported DNA cross-linking polymer gels. We talked about how important is stress relaxation for biological tissues. So we also want to tackle this challenge. As we know, other system has difficulty to change stress relaxation without touching other parameter. In DNA world, it's known that duplex DNA, the mechanical stability is sequence dependent. The longer the sequence, the higher the force you need to rupture this two strand apart. So we designed stress relaxation crosslinker, which essentially changing the length of this overlap domain, which is the weakest part of the crosslinker. So you can imagine that 18 nucleotide overhand domain will have higher stability and therefore more stable, slower relaxation time than the one that only have 6 nucleotide. By using different lengths of the crosslinker, we can change the stress relaxation time from more than 10,000 seconds down to sub-seconds. This range nicely cover the typical stress relaxation time in the biological tissues. Also, the big unique part of this strategy is that we can change the stress relaxation time without changing the stiffness. So it's independently controllable. Another nice property about DNA hydrogel is its self-healing. Since DNA duplex binding is reversible, we can break the gel, and break the bond here, under mechanical shear force, basically liquefy it. And on this plot, in this experiment, we basically torture the gel for multiple breaking and recovery cycles. And you can see that in the resting state, the G prime is dominant over G double prime, but at the breaking phase, G double prime shoots up and G prime goes down. And you can do this for like 10 cycles, but at the end, if you give enough time, the stiffness can re-establish to its original state. This self-healing is particularly useful in extrusion printing. You can think of when the gel was squeezed over a thin nozzle, it undergo a strong shear force. And in this case, the gel could be liquefied when it was extruded out, but once it's out, then it will regelate it and form a gel. This is also more compatible with the cell, since that the cell, the strong force can lead to cell death if you don't have a liquefied structure here. And self-healing basically protect the cell from the strong shear force. So one can also do bioprinting on a sophisticated bioprinter to create a tissue-like structure like this grid for a more realistic tissue structure culture. So after all of this amazing property, now it comes to the cells part. As the cells comes in, we ran into a couple of problems. First thing is the temperature. The cells couldn't withstand temperature higher than 40 degrees. So when we do this annealing of DNA crosslinker, we cannot just heat them to 95 degrees. The cell will die. And in the beginning, we just thought, okay, let's just mix 2 solutions. And this 2 solutions have in the splint strand and the complementary splint strands separately. And we just mix them together very fast. But the problem is that the rate of crosslinking is extremely fast. So if we mix these 2 solutions together with the cell, even with pipetted very, very fast, we still couldn't make a homogeneous gel. So what it ends up is a very non-homogeneous gel structures, and the mixing is very poor. So in this case, you can imagine the cells sitting at this region will feel a completely different mechanical property than the cells sitting here. So we propose a solution inspired by the matrigel gelation mechanism. For people who have worked with the matrigel, you keep the matrigel. The matrigel is liquid at 4 degrees, and you can mix with the cells in the medium at this temperature on ice, and then you drop it into a cell culture plate, put it into the incubator at 37 degrees, and it starts solidifying, form a gel. And we thought, oh, this is a very convenient features. Can we do that with DNA? So we developed a so-called heat-activating crosslinker. By adding a blocking strand, X, at this overlap domain, at low temperature, they're metastable. They won't fall. So at 4 degrees, when you try to mix 2 solutions, they won't start cross-linking. They won't form a gel. But as soon as you put your mixture into 37 degrees, this blocking strand is not stable enough to bind at all. It melts away. So now after you have properly mixed all your components for cell culture, you put it into the incubator and then start forming cross-linked. This allows us to form a homogeneous gels for better mixing. And also better mixing means the gelation can be complete and we have improved gel stiffness when we have the blocking strand. You see this plot when we keep it at 4 degrees and heat up to 37 degrees. Without blocking strand, they start randomly binding to each other at 4 degrees. But this mismatched DNA sequence will quickly melt at elevated temperature, so the stiffness basically goes down. However, with the blocking strand, it is still liquid at 4 degrees, but once you heat it up, they start forming a gel, and it can reach a theoretical stiffness value. This strategy was later used in all of our cell culture experiments. So yes, we started with some cell viability tests with mesenchymal stem cells using this heat activating strategy. And we can demonstrate that over a week of culture, viability of cells is high, comparable to the matrigel references. And on the right side is basically the life-death staining result that shows nicely that cells are sitting in 3D and showing high viability, shown in green. The next problem we ran into is the stability of the DNA hydrogels. There are plenty of nucleases that would eat away your DNA in this typical cell culture serum, fetal bovine serum, FBS. In this video that we showed that if you just put the medium on top of it, your DNA gel will basically be digested away and just disappear within 24 hours. Luckily, well, unfortunately, this has happened to other DNA gel study as well. That's why they couldn't get a success. But luckily, there are many ways to counteract these digestions. One of the solutions that we end up using most often is adding a nuclease inhibitory agent. In this case, we use actin, which is a natural inhibitor of DNase 1. By adding actin into your medium first, before adding to our gel, let's see this video on the left side again, it gets eaten away. But if you inhibit the nuclease activity, the gel can be protected for a couple of days. This solution is not perfect, it's not 100% protections. So right now we actually prefer to use serum-free medium, a synthetic defined medium for cell culture that allows us to culture our cells for up to weeks without any problem. But looking at this, it's actually a feature, not a bug, because now we can release the cells by digesting our gel with DNase, and even controls the degradation rate by adding a different amount of this inhibitory agent. In this plot, we supplement different amount of actin in the solutions, and you can see that the rate of digestions goes down at different times. And we also utilize these features to release the mesenchymal stem cell from our gels after 9 days of culture. And the cells are still alive after releasement. Now I want to show a couple of highlights that we have achieved in the cell culture part. We have tested 3 different types of cells in our system that show successful culture in it, including the kidney cyst cells over here in collaboration with Alf Honigman lab in MPI. Induced pluripotent stem cells in collaboration with Kostevaner in IPF. And also, we tried out organoid culture, this placenta organoid in collaboration with Claudia Gary. I will take a quick dive into each of them with you. First of all, this MDCK cells start growing starting from single cells and into the cyst-like structure. And the unique part is this cyst has a polarity. There's an apical in and apical out polarity. Also, we want to highlight that for cell culture, we functionalize our polymer chain with RGD peptide, a cell adhesion peptide. for the matrix to interact with the cells. And you can see that how important is this cell adhesion peptide. Without the RGD peptide, you get a cyst that is almost apical out, which is like this is in vivo. Apical is in vivo natural state morphology, and apical out is abnormal. And without RGD, they couldn't sense any of your matrix, so they just flip its polarity. And by adding RGD, we can start seeing some cysts start to form this correct apical in polarity inside. However, we can still see some of the cysts did not form this native correct polarity. And we believe that the gel mechanics, the viscoelasticity plays a role inside the polarity as well. So Tim in our lab has been focusing on how to change the viscoelasticity and how does it regulate cell polarity. For the iPSC study, we show that the induced pluripotent stem cells grows into a similar morphology as in matrigels. And look at this structure, is very roundish, very nice. The next step is that we want to know how viscoelasticity affects stem cell differentiation, because a lot of study was done on iPSC to study differentiation into different lineage. And we want to use this softness of our gel to cultivate this kind of cells and to see what viscoelasticity play in role. Oh, this is just a cool picture I want to show to you guys. This is an iPSC cyst, and you can see that they grow from a small cluster and expanded gradually and push away those matrices. So you have this hollow inside, well, a lot of liquid inside, but there's no matrix, and matrix was pushed against to the outside. The reason why there are so many backgrounds is that it's just a dirty staining, a dirty immunostaining. And also this gel has a higher DNA content. And so sometimes with higher DNA content, we will have a background signal from the stoppy staining, a nucleus staining. And so that's why we see this Schlier, and it's a very cool image over here. The last is the organoid culture. This placenta organoid was used to culture in matrigel always. And it's the first time that placenta organoid can be successfully cultured in synthetic matrix. And we showed that we can culture up to 21 days, including several cell passaging steps by using nuclease. We showed that even after several passaging, the cells has a high viability and a morphology that's also comparable to what is in the matrigel. But in matrigel, they form a wrong polarity. Also in our gel, they show a wrong polarity. So right now, what we want to do is, we want to explore what is the optimal mechanical conditions for them to grow into the correct polarity that mimic the placenta polarity in vivo. Finally, the last part, I want to highlight that the DyNAtrix, we call our DNA hydrogel called DyNAtrix, stands for Dynamic DNA Cross-Linking Matrix. The DyNAtrix-based culture is highly adaptable. The general workflow looks like this. You have two precursor solutions with heat-activating cross-linkers. You mix with cells in your medium together with these two solutions at low temperature. Then you put them into the incubator for 30 minutes and form a gel, just like what you do with the matrigel. And during this culture, you can do imaging. We are also interested in switching the mechanical property of the gel live, so we can monitor the cell behavior change by live-cell imaging. And we're also interested in upgrade of material, for instance, with DNA force probe that can sense and track the force between matrix and cells in real time during the development. We're also interested in adding DNA aptamer, which is a unique DNA sequence that can bind to certain molecule like growth factor. Then we can study how individual growth factor affect cell behaviors. After you have done your culture, it takes your image. You can also release your cell by adding nuclease. And the cells can go to do, you can use this cell for downstream analysis like FACS, immunostaining, or single cell sequencing, etc. But you can also just passage the gel, re-embed it into the gel, and start your culture again to repeat your experiment. I want to highlight that we're able to keep the material cost comparable to matrigel. Right now is 25 euros per milliliter for small scale synthesis, and we expect to even make it cheaper in the large scale productions. Our material design has been patented, and we also are interested in creating startup in the future. To summarize, that DyNAtrix is a fully synthetic, programmable, and versatile matrix for 3D cell culture. The DNA sequence allows us to determine several different properties that we can create programmable biomechanics, allows us to mimic matrigel to have this heat-activating gelations, and we also show that it's compatible with several different cell types. And for future, for manufacturing, you can also do bioprinting. I think I've spent a lot of time talking about the project, and I would like to spend the last few minutes introducing our team. Our group leader, Dr. Alicia Krieg, has obtained his PhD in chemistry in the Weizmann Institute in Israel, and he has done his postdoc in Boston, in Harvard University, at the William Shih Lab. He was also now in Dresden in IPF, and he was selected as a young investigator since 2021. We have very diverse application on this material platform. Right now, my project focuses on how we engineer this material platform for multiple purposes. My colleague, Sarah Speed, has used this material platform to create programmable nucleic acid and biomolecule capture technique for next generation sequencing. Krishna Gupta works on his PornoCare project using a special gene switch called Y switch that he developed for a better diagnosis device. And Tim, the one most closely working on this project with me, he's a cell expert, and he now works on how viscoelasticity changes cell behavior. So you see that our team is quite international, and everyone brings diverse set of skills to the lab. Yes, I just want to quickly walk through my academic journey. I have done my bachelor thesis in National Jiaotong University in Taiwan. Well, now it's Yangming Jiaotong University. In the lab with Professor Eric Huang. I have a really great time there, working on neuron, the dynamic of cytoskeleton and neuron cells. He's also the one that supports me, encouraged me to go on a semester exchange in Germany, where I met another referee, Professor Ralph Kemkema, after I joined his lab doing a student project with him. Both of them have become a strong support for me to later study in the master program in TU Dresden. During my master, I've done a couple of lab rotations, and I also work as a working student and done my master thesis with Dr. Alicia Krieg. That's how we connect. And since we made some good progress in the project, I decided to do my PhD with him, and this journey will be continuing. The last slide, I want to shout out to my whole group, and also all the collaborators we have share among the Dresden Institute's community. Our group is quite active. We have done several scientific outreach, like going to high school, showing students how molecular biology can be used in real life. And we also joined the city run for every year. Now it has been a tradition to collect 4 medals before you can graduate. That's the end of my presentation. Thanks, everyone. I will go back to the summary slide. Maybe stop there for people to ask questions.
好，感谢 Jasper 今天就是很丰富的分享。那现在是 Q&A session， 然后这时候其实大家就可以打开自己的麦克风，然后建议就大家可以先自我介绍一下，也可以讲一下自己的背景是什么，然后再提你要问的问题，这样子。
我可以问问题吗？问2个问题。
那 OK，
嗯
等人请。
好啊，我叫耿源不知道看不看到我，就是我在 Penn 当博后，就是做那个，我之前是做 bioengineering，那现在是做那个cell biology，然后 Penn 有很多人做这种就所谓的 stiffness 的 work。那我
嗯
根据你的 talk，我有两个比较其实蛮直接的问题吧，就是说你刚刚说之前的系统，就是之前的那些纯 DNA 的胶啊，就是他们会有一些 immune 的。那你说你想要降低这个 immune response，说你降低了一些，就是你的系统有一些 advantages。那但是我在后
嗯，
面的 slide 好像没有看到，就是你就
哦，
是
嗯哼，
这两个 comparison，就是你有没有发现说你的系统真的有降低一些 immune response？那你怎么做这些？因为我不是做 immune 的，所以我不是很清楚。好，今天先问第一个，然后之后我再
嗯，
问第二个。
就第一个问题就是 immune response 怎么降低，跟我们到底有没有 validate 它到底有没有？
对，我意思是说，
对
你说你比之前的好嘛，就是理
对对，
论上，但是实际
嗯
上你怎么去做这些测试？
嗯，是我没有秀在我的 slide 上面，但我们有去做那个 blood compatibility test，去测说会不会 trigger blood coagulation 跟那个 immune cells 的 activation。然后我们测出来是几乎是没有，就跟那个innocent 的 material 的 control 是差不多的。嗯，然后要应该说降低 immune response，我们有刻意做一些调整，比如说像某种特定的 sequence，像 CpG DNA sequence、CG 这种 sequence，它很容易被toll-like receptor 去 recognize，然后就会 trigger immune response，所以我们有刻意避掉这种 specific triggers，那个 sequence 那个会被 cell receptor 给 recognize 的。然后 so far，因为我们用比别人还少的 DNA，所以这个已经就是一个先天的优势嘛。So far 我们没有看到acute immune response happen，但我们还没有真的 culture，比如说 macrophage 或者是一些 white blood cell in our gel 去 further confirm this。Yeah. 然后你说你还有第二个 question 对吧？
对，就是我可能漏掉一个部分，就是说你们那个 hydrogel 的那个上面是有什么 molecule 吗？不然细胞怎么会贴上去？是有什么 integrin
哦，
receptor
对，
那种 molecule 吗？
我们有加那个 RGD sequence，我看我
哦，
能不能 pull out 这个，不知道 pull out 这个 paper。我们在那个 polymer backbone 上面跟我们呃 anchor 加那个 anchor 选 DNA 的 strategy 一样，我们也是把 RGD peptide 加进去，然后一起 co-polymerize，
嗯
所以那个 backbone 上面就有DNA 跟 RGD peptide 同时在上面。
嗯，
对，
那
然后那
OK，
个 RGD peptide 的作用就是
嗯
它会跟 cell 有 mechanical linkage 嘛，然后 cell 就可以 sense trigger 的 mechanical signaling 这样。
嗯哼，
对，
那你那时候在看，抱歉，我再多问一个，就是你那时候在看那个
没问题。
survival 的时候，那
对，
个它的 cell cycle 有被影响到吗？还是就是至少跟 matrix 的表现比起来，还是跟那个
我们一直都
力同学？
你说 cell cycle
就比如
proliferation，
说，对对对，是不是长得比较
嗯
快，或是还是有长得比较慢之类的？
嗯，我们发现好像 matrix 长得比较快是它的趋势，就是都是有这个趋势在，这 matrix 它长比较大颗，然后
你说跟 2D 比吗？还是跟你的系统
跟
比？
跟我们的 gel 比，就是 Match Gel 跟我们
啊，
gel 比起来，我们有
嗯
发现，在 Match Gel 他们通常长得比较快、比较大个，尤其是在那个 iPSC cells
，OK。
尤其明显。这样，对，
哦，我问这个问题是因为你刚刚有一个 slide 是秀那个 viability 还是什么 survival rate，那我只是想说你那时候是怎么控制 vi 就是这个东西，因为如果它长得比较快的话，那你可能要做一些 normalization 啊之类的。没有，我只是怀疑说是
嗯，
不是怀疑啊，就是
哦，
疑问说大家怎么去做这件事情，因为我不是还没有做到这个东西，所以想问一下大家怎么做。
哦，我们其实就很简单，就是把那个这两种带，就是 life
嗯
带、dead 带这两种加进去，然后去数它的 percentage，就是有多少是红色的细胞，就是代表它已经死了，然后绿色
嗯，
是活的细胞。对，
对。
嗯，
不过
是
overall
个
你都可以
蛮
看到，
basic 的。
你都可以看到那个它长成一个圈嘛，就是那个 cyst，基本
对，
上 OK，那就是
对，像某些细胞它会特别长成一个 cyst，不过
嗯哼
像MSC 这里我们就没有看到这样，它就是一个
嗯。
single cell。
嗯至
对，
少那个 result 看起来蛮 striking 的。好，谢谢。
嗯。 然后我们好像有一些问题是不是在 chat 里面还是没有？
应该是没有，对，那 chat 应该是刚刚我
好，
们在宣传一些事物，然后对，然后不知道还有没有人有相关的问题，因为我知道在场也有人做 IPS 的，然
嗯
后对于你们来说，可能你们只是拿材料来应用，然后或许你们自己在做这些 culture 或应用的时候遇到什么问题，也可以这时候问，因为他们
嗯。
是做材料的，所以他们可能对生物到底在 culture 上 technical 会遇到什么问题，可能没有想到，或许你们可以提出不一样的想法。
Hello，听得到吗？
可以。
可以。
我这边也可以问一个问题。我是永成，我可能最近才刚加入 Tyra 的这个会员，所以然后我也不太了解材料，但是我觉得你做这个用 DNA 的这种 dynamic 的特性来去改变材料的那个 property，我觉得很有趣。然后我的问题是关于就是你在 gelation 之后还
嗯
有没有机会？就是在 include cell，然后 gelation 之后有没有机会再透过加入一些 polymer 或者是 DNA，然后就是再进一步改变它的那个物理特性、材料的特性？因为说不定细胞在比如说它的从 single cell 然后变成整个 tissue 的时候，它说不定
嗯，
需要不同的就是 microenvironment 的刺激嘛？
对
有没有这个
对。
可能，就是透过 DNA 的这种特性，然后进一步的去就是 engineer 它？
嗯，其实我觉得这是一个很好的问题，因为我们 group 现在就在研究这方面，就是我们可以去改变这个 DNA-crosslinker 比如说 mechanical stability，尤其像在这个 kidney cyst 这个研究里面，Tim 他就去 design，他做了一种叫 switchable stress-relaxation crosslinker，就它可以加入另外一个 strand，然后把这个 stress-relaxation 的 property 改变，从慢到快或快到慢都可以。然后他去做 lifestyle imaging，就发现，哎，真的那个 polarity 就这样 flip，我就 flip 过来，这样去改变。所以确实是可以用 DNA nanotechnology 去改变 in situ 去改变这个 gel 的 mechanics。
哦，所以是就是已经 gelation 了，但是你从外
对
面再加其他的 crosslinker 或者是它的就是
对
叫什么 competitor，也是可以把里面的
对
crosslinker 替换掉的。
对，你可以把它替换掉，然后只是
哦，很
reversible
酷。
这样，就它替换
嗯
从快到慢，它可以再接着把它从快到慢，慢到快再回去这样。像
嗯
他做的话，他好像是在第4天跟第7天去改变，然后就都可以达到一样的效果这样。
嗯。
嗯
那我能不能让我再加问你？刚好你换到这一页，就是你提到的这个 RGD，就
嗯
是你们在选这个要放什么 peptide 的时候有什么考虑吗？因为理论上，比如说像 matrix 它应该有很多不同的就是 cell adhesion 的 molecule，就你们在选 RGD 这个 molecule，以及你们选那个就是 organoid 的应用的时候，你们会有什么考
嗯
虑吗？比如说你们为什么是从 iPSC 开始做，而不是做其他的，比如说更 advanced organ 或者是更 complicated 的 model？
嗯，我们先回答第一个问题，就是那个关于这个 cell 的 adhesion peptide 的。我们当初选 RGE 单纯只是因为它是一个最 commonly used 的 peptide，而且它是一段 laminin 的 sequence 嘛，然后 laminin 是 matrix 里面最 enriched 的一个 component。所以我们就优先选它。但我们也知道 matrix 里面也还有其他像 collagen，然后有一些其他 synthetic peptide，别人有在用，比如说什么 G Foger 或者是 ECAV、IKVAV 这种。然后我们现在就在尝试 synthesize 不同的 peptide，在那 polymer 上面 synthesize 不同的 peptide，然后想说去看，如果我们 mix 不同的 peptide 会不会有让细胞长得更好，或者是对，这是我
嗯，
们目前在做的。然后，你刚刚第二个问题我忘记了。
就是你们在选，比如说你这边展示的几个 organoid 的不同应用的时候，就是这个 model 是怎么选的？比如说你做 iPSC，或者是做刚
嗯哼，
才是 kidney organoid 吗？
哦，这个是
还是
endothelial cells，这个可能
嗯，
算一个对。不
就你
然你是问
们在选这些 application 的时候有什么考虑吗？比如说这些是
哦，
原本就有在 Matrix 9里面，就我不是很清楚，但是
嗯哼，
这些是原本就有在 Matrix 9里面培养的，然后你们把它拿过来用在你
嗯
们的 g 里面，看能不能就是 develop 成一样的 phenotype，还是
嗯
对我想问的是这个
嗯，
方面。
这3个都有在 matrix 里面， 就是一般他们原本standard 的 cell culture 都是在 matrigel 或者是在不同 concentration matrigel 里面养的。然后因为我们的 softness 跟 matrigel 是很像的，所以我们想知道我们能不能 recreate matrigel 里面这个 morphology 或 even better。比如说像这个 kinetic， 我们知道它的 polarity 是跟mechanical property 有关系的，所以这是在 matrigel 里面你很难去调整的 mechanical property， 你必须改变那个 protein 浓度去调整。所以我们想知道我们可不可以用比较 control 的方法去调整。然后 iPSC 跟 placenta organoid 一直以来都是在 matrigel 里面培养，但是我们想看说我们能不能利用我们这个很特殊的programmable biomechanics 去 direct 它的，比如说像 iPSC differentiation， 然后像 placenta organoid 它的 polarity 在 matrigel 里面根本就是错的，就是你没有办法制造出一个对的 polarity。那我们想知道我们可不可以用我们的 gel 去了解到什么样的 mechanical properties 才能制造出对的 polarity， 这样。对。所以我们一直都是，然后选这些 cell，大部分原因都是一些 collaborator， 他们都很有兴趣，他们其实一直都想要踏进 synthetic matrix 这个世界这个领域，但是他们一直找不到一个对的材料，或者是可以 compatible 材料，然后刚好我们有这个相近 softness 的材料，然后让他们去做测试，这样。对，
了解，谢谢，这个真的很酷。
不好意思，我再问一个，根据刚刚的问题，我刚想到一个，就是你刚刚在看那个 matrigel 跟这个你的 gel 的比较的时候，你们那个 double prime G prime 跟 G double prime 都是类似的吗？还是其实某个部分有一些差别之类的？我是说，如果你在看那个 proliferation，你刚刚说你的系统是会 proliferate 比较慢嘛，对不对？那我在想说，因为我听说有人说 stiffness 会让细胞 proliferation 变慢，但他们就modulate stiffness，然后做这些实验。然后我只是想说，如果你的系统的那些差距是不是因为这些 mechanical property 造成的，还是其实是一些 chemical property 造成的？对，我只想说你有没有这样的方面的一些评论，这样子。
其实这个跟 Tim 他现在正在写的 manuscript，他应该可以很好的 answer 你的问题。就我们现在从我们的 result 看来，你的 stiffness， 还有 stress-relaxation， 还有你的那个 cell adhesion peptide density， 对于这个细胞 cell development 都有一个影响。然后，如果你有个很高的 stiffness， 但是你却不 stress-relax 的话，那个细胞很难长大。因为它就是没有空间可以 expand 到外面。但如果你high stiffness， 然后有 fast relaxation 的话，我们有看到它的 size 就变大，然后尤其在 soft，跟 fast relaxation 的 combination 它长得最大，所以确实这个大小它是跟 matrix 它本身的 mechanics 是有关系的。然后 matrix 本身其实是算是一个中间偏快的 relaxation 的 matrix 这样，然后再加上它有很多很多很多的 cell adhesion peptide， 所以就有更高几率去 trigger 这个 mechanical signaling 的 pathway， 所以它可能就更多细胞有机会去一直增长这样。
好，谢谢。
那我觉得我们的 cell adhesion 的 density 可能没有 matrigel 这么高或这么 enrich， 像你讲它有很多不同 component 在里面 ，matrigel 很多不同 component 在里面，而我们只有其中一种。所以我们也想知道说，到底这个 ligand 的 diversity 跟 density 有没有影响？这样。
好，谢谢。
我也可以补充一下，就是像我自己是做 cancer， 我也用 matrigel 就是从老鼠去抽出来嘛，它每一批 batch 其实都会有影响。然后我可以想象 Jasper 的这个系统，应该它的那个 QA/QC 的部分就是比较好 control， 因为它毕竟是用一个合成的方式，而不是从就是动物或者是细胞去 extract 出来这样子。对，那我好奇一下，就是最后问一下，就是你们现在就是在讲 organelle， 那像我以前做 cancer， 我们也常常用 matrigel 做一些 invasion 跟 然后我们也知道 matrigel 或 collagen stiffness 会去影响 cancer cell morphology， 然后 either 是 benefit 或者是有 drawback when it comes to migration or invasion。你们这个有想要去尝试做这一块吗？去 embed 不管什么细胞，然后来了解它的一些比较 dynamic 的移动啊，干嘛之类的。
就这个方向，我们其实一直都有在思考。 因为有很多 collaborator 做 cancer 的，他们也想尝试看看 synthetic matrices，然后也想试试看这个 metastasis 这个 pathway，能不能用我们的 gel 来 study。不过我们目前看来，我们的 gel 长这种 cyst 的 cell 长得最好，就长得比较好。然后 single cells 那种 migration，我们目前都还没有看到有任何细胞可以成功做 migration 这样。我们有一些 hypothesis 说为什么这些 cell 不 migrate，
哎，
就是
可是你们有加 chemoattractant 吗？如果没有 chemoattractant，它当然就不会跑啊。
我们之前试过那个 Huvex cell，blood vessel cells，然后如果你加那个 VEGF 进去的话，它就会开始变得很兴奋，然后就开始到处跑来跑去，然后 form 那个 vasculature network 嘛，到处 connect。但我们目前都还没有办法成功让他们到处这样跑来跑去，然后 form connection，这样就不论我们这样 dump 多少 VEGF 进去，所以我觉得有一个 puzzle is missing，就是有一块是 missing 的。
那我想要问，你们有办法一片大胶，然后做一个 gradient 吗？例如从硬到软，然后做一个 gradient，你们是有办法控制这样子的？
这也是一个 idea，我们觉得很酷，我们也想尝试。In principle, yes，我们可以做这件事。然后一开始我们的 idea 是想说用一个 like a photosensitive molecule，然后这个 photosensitive molecule 可以去打断这个 DNA，让你可以 soften 你的 gel，然后当你照那个光上去的时候，它就会把这个 DNA 给炸掉。然后我们可以用这样去 pattern our gel。我们还在找一个适合的带，因为之前我们认为可以 work 的带似乎不太 work，就我们不管怎么照它，好像都没有把它打断这样。
了解。
但
会这样问的原因是因为就是 cancer 从 primary 到 secondary 在移动的这个过程，其实比你想象中还要刻苦啦。就是它的 surrounding environment 硬啊软啊硬啊软，它必须要真正能成功的 cancer cell 其实很少，但是会成功的那个 cancer cell 就会变 aggressive，所以当它 spread 出去，为什么我们就很难去救这个病人的关系？所以大家就会想要知道说，哎，那有没有一个 in vitro system 可以帮助我们做一个 real time tracking？然后在软的状况之下，什么细胞它的移动模式是比较可以硬的时候是什么？但是现在就是大部分的系统就是很单
对我
一啦，就是就是在硬的或者是软的，然后你就一些 morphology change 大概就这样子，对不对？所以如果能把真的有办法做一个 gradient，然后可以是 switch over time 或什么之类，然后可以有个 real time 观察，我觉得对做 cancer 的人应该就是他们会蛮有兴趣。
这个的确很酷。就是 in principle， 我们也可以就是做那个 bioprinting 嘛，就是你把不同硬度的 material printing into certain patterns，这也是有很多方法可以做到这样。只是我们想说很 conveniently 嘛，你用光是这个非常 precise 跟 convenient 的方式，你可以你就放个 mask 在上面，也可以印出不同的 pattern 这样。对，we are working on it
好，那不知道还有没有什么问题？不然如果没有的话，因为时间的关系，可能我们就可以今天这边结束。但是我想 Jasper 也可以留他的联络方式，或者是我们的报名，就是我们的报名官网那时候也有附上 Jasper 的 LinkedIn，大家就是也可以欢迎试一下跟他 connect，然后或者是未来还有什么就是像 scientific 相关的问题想要问他也可以，或者是说想要有人如果是在台湾想要去德国留学。或了解 Dresden， 像 Dresden 如果对这个有兴趣的话 ，Dresden 这个 cluster 对 mechanical biology 算是在学界蛮有名的 ，biophysics 或 mechanical biology， 我们那边就是那个 cluster 就做得蛮好，所以如果想要了解的话，都可以试一下，就是联络 Jasper。另外就是别忘了，我就是在刚刚 chat box 的地方就是有放一个 survey， 然后呢就麻烦大家等一下可以活动结束之后填写一下 survey， 然后我也会把survey 的大家的 comment 或者是有什么想法，如果有额外写下来的话，我都会就是跟讲者说这样子。
好，感谢你。
好，那我这边先 close my
我快速的把我的 LinkedIn 放在 chat 里面好了。
好，好，那我这边先 stop recording。